let's start our, so we have now 2 hours of discussion. The first hour will be a discussion panel of the 3 sessions from this morning. And then the last hour will be a discussion of how to create an exRNA data analysis challenge. So I think for the questions, I'm going to try and give each of the 3 sessions 20 minutes and do them in reverse order. The last session is fresh in everyone's minds, and the deconvolution session sort of leads into the discussion of a data analysis challenge. So Boyan, some questions for you. Are unannotated RNAs the same as unassigned RNAs?
Yeah, I mean, it really does blur the line. We found that unannotated RNAs can emanate again from intronic sequence. It can, you know, scratch against really the splicing surveys of mRNA. So if you have very rare isoforms of mRNA that aren't fully characterized, then you know these intronic regions aren't properly characterized themselves. So there's a lot of overlap, I would say, between unannotated and unassigned.
And which kit did you use for generating libraries of exosomal RNAs for RNA-seq?
Okay, that's a good question. I think we used.
Boyan, you want me to answer?
Navneet. Yeah, go for it. Navneet, yeah.
We did the total small RNA library, and we polyadenylate everything that we can, the small RNAs, and then we prepare the library and we sequence them.
Let me move to some questions for Leonora. Which class of RNAs were mostly present in EVs from responders versus non-responder patients? I'm not sure, is Leonora available? Maybe not. Let me scrounge around a little bit and find my questions for Elizabeth. Actually, I think we asked, we were able to ask Elizabeth all of her questions because she had some time at the end. So let me move on to the RNA binding protein section. Someone asks, does ERCC2 aim to build an RBP atlas associated with exRNAs?
I don't know that that is exactly one of the goals. We're definitely, I think, approaching that with the release of these intersects between all of the ERCC stage 1 data and all new data to come in with these RBPs, as well as the information and validation that Bogdan's group is doing as well. I think Alex has a comment.
Yeah, building on what Emily just said, there are actually 2 parts to the answer. One is we are moving beyond standard annotations that are currently in the atlas and allowing for flexible analysis of any sets of regions of interest. For example, ENCODE defines RNAs bound to RBPs. We would like to allow atlas users to access any sets of such regions, whether currently annotated or not. So we are adding that flexibility that will help study, RBPs, but any other class of, RNA carriers as well. So that's one part of the answer. Second is we are going to annotate specifically, RBPs, and this is what Emily mentioned, and make this data available as a matrix of numbers where somebody can just pick their own RBPs and analyze them. But this is not really a new atlas. It's more of an extension of exRNA atlas and new feature. So we'll be doing that. And the third part of the answer is RBPs are complex, right? It's not just RNA in them. And we are building kind of an umbrella atlas that will include extracellular vesicle and particles, not just from the RNA cargo point of view, but also protein point of view. This is what ERCC2 is about. We're including flow methods, single vesicle characterization methods, antibody characterization of carriers and so on. So we'll be able to intersect, both the cargo and the carrier using this extension of the exRNA atlas, which will be more of an EV and particle atlas, like an umbrella integrating carrier as well.
Just to add one more point, we are planning to release the single knockouts profiles for each of the RBPs, which could be useful for people doing signature-based deconvolution, running different deconvolution or algorithm, and also to identify which, for example, for some of the people just presented, identify the dependency of specific fragments upon a specific RBPs.
Someone asks, can I get RBPs from ENCODE? This may be for Eric, if I just provide differentially expressed small RNAs.
So that's not something that I believe there's an existing tool for.
So that's something
Yeah,
they'd have to figure out for themselves, like write the
Yeah,
algorithm.
figure out for yourselves, or I mean, if you reach out, I can try to connect you. There are a couple of resources that are in different stages of being released where you can ask, for example, this is the gene I'm interested in, and what are the RBPs that have the strongest binding to that gene or that region. I have a couple of different people building tools to browse data like that, but nothing that's like upload a gene list and do that kind of analysis right now.
Another question for Eric, and it's not quite data analysis, but do you have any thought about how to adapt your experimental protocols to work in biofluid derived samples?
Yeah, I was talking with Alex about that a little while back. There's definitely some challenges, but in general, I think it would work fine. I mean, because all of that, everything is IP based. So essentially, the challenges are, we generally use quite a bit of material, which is really just because some of the steps are somewhat inefficient. We've tried to make those better, but especially cross-linking is still quite inefficient. And so what we found is there are certain RBPs that you can take some shortcuts and get much higher yields, and the data quality does not suffer. AGO2 actually being one of those. So I think that's the direction we've gone for, not for extracellular RNAs, but for some other things where for certain RNA binding proteins, we can push things quite a bit further. And I think for extracellular RNAs and vesicles and things like that, I think that's sort of the direction to go. It would be hard to get enough material to do the normal clip procedure, but for certain RNA binding proteins or certain tags where you can optimize the conditions, I think it should work reasonably well.
Thanks. It may be early for people to have a good answer for this question, but I'll ask anyway for all of the RBP folks. Did any of the RBPs you found have multiple functions in cells? I think the default answer is they do so many things. They're all crazy deep in the nucleus doing splicing kind of stuff. But
yeah, I mean, we've definitely seen, and admittedly, most of what we saw matches things that were in the literature. So there are a couple of RBPs that we looked at that clearly have some splicing regulatory roles and also some roles in regulating RNA stability in the cytoplasm. We've seen some and this is also well known that some of the splicing regulatory RBPs also then seem to have effects on RNA export from the nucleus and things like that. So that's definitely true. I would say the one thing that we didn't see very much of, although this may be because we were doing 2 cancer cell lines for ENCODE, is we didn't really see very much of evidence of RBPs that had different functions between the 2 different cell types. So I think that may be something where you really do need to go more towards different tissues or different conditions to really see, really different roles for the RBPs between different cell types.
Sorry, for RBP exRNA complexes that are released by active sorting, can they be incorporated by specific cells? That's another biology question.
I think that's an excellent question that no one's really looked at. I think it's the same idea we have for vesicles, right? Is that they're released and taken up by other cells in some manner and theoretically functional. But as far as I know, no one's actually looked at those that are released outside of vesicles go through the same process. We're kind of assuming that they do. I
have I guess a follow-up question. Don't we know something about Argonaut? I'm just curious from the panel. We know what Argonaut does in the cell, and we know that there's a lot of Argonaut in biofluids. Do we know anything about mechanisms, how it's secreted, whether it's taken up by target cells, and what it's doing, if anything? I feel like that might be the most understood protein at this point.
I can answer a little bit on this. I mean, we know that Argonaut is recycled by membrane-based mechanism in cells and different endosomal sorting. So that could be one connection to EVs-based secretion. Although this is quite minor compared to the amount of Argonaut secreted in cells in biofluid. What I believe is that the protein is quite stable in contact with microRNA and would accumulate upon cell lysis. Or I don't know if there is any specific mechanism leading to secretion. I think they are just very stable. But
so it might be a case like Juan Pablo Tosar discovered with tRNA fragments, that it's what's stable which is left in the biofluids after all the enzymes do their work.
Yeah, we also have. I mean, we never had managed to prove it with experimental setup. But what I mentioned during my talk, that the fact that RNA granules could also stabilize RNA in biofluids is also one hypothesis, because when there is phase transition and aggregation of these RNA binding proteins, they are also more stable and they could be also released this way through different mechanisms. And notably, we know that there is ways for the cells to degrade RNA granules, and they could be exported out of the cells, maybe in some instance. And these RNA granules, which contained a lot of Argonaut and also different proteins, and they could be then digested by extracellular RNAs and then dissociated into single Rbps.
Thanks, Bogdan. Another question, what was the size of tRNA fragments in the urine RNA RBP complexes? I don't remember who this would be directed to. Perhaps someone who is not here. Here's a question for Bogdan. Is there an effect of silencing of RBPs on transcript isoforms in extracellular RNA?
So silencing on the RBPs on transcript isoforms in of the extracellular RNA.
Mhm.
Well, I wouldn't be able to answer this because we looked at small RNAs mostly. We didn't perform any long RNA-seq. Although this would be the next step. I think if you ask Eric, there is very interesting impact of some RBPs on splicing. I think they conducted a number of knockdown experiments. Maybe you can comment on this.
Sorry, maybe I think I missed the question.
Bogdan, would you mind restating it? You probably understand it better than I did.
If I understood well, the question was whether certain RBPs will be involved in splice junction, like splice isoform selections, and whether this could impact exRNA profiles.
I see. I mean, so the latter I can't answer at all because we frankly didn't look at that. The former, yes. I mean, so there's definitely quite a bit of splicing data and code to look at, and that's one of the interesting things that we've seen is that some of the spliceosomal components, you know, some you see association at almost all splice sites, and some you see very specific patterns of enrichment for different spliceosomal complexes, so different stages of splicing. And we've been able to show that you can sort of, well, this is something we're doing ongoing work on, but you know, at some level that seems to correlate with introns that are sort of either more slowly spliced or the recognition is somewhat differently regulated from sort of a constitutive intron.
Here's a question for Emily. Using the exRNA atlas data, does using the exRNA atlas data put a bias to small RNA binding proteins? What about mRNAs?
So
It's sort of related.
yeah, we essentially take in samples that have been like small RNA-seq has been performed on these samples. So yes, we would expect biases towards these smaller RNA species. But we are also obviously seeing fragments of these mRNAs and some of these longer RNAs. So I believe someone has been looking at taking these fragments of longer reads and basically putting them back together to measure if you can get like mRNA reads. But at this point, the atlas is really focused towards small RNA sequencing. So that's probably mostly what you're going to be finding.
Yeah, just for those new to the field, when the ERCC was founded in 2013, the first several years really focused on small RNA-seq and a lot of focus on microRNA because people knew what microRNA was doing. And so the field is just sort of ramping up into also looking at long RNA-seq. And it's too bad that Elizabeth wasn't able to provide commentary during this panel, but it's a bit newer in the field.
I would also like to add that yesterday's discussion about modification, phosphorylation, hydroxylation, right, is relevant here, right? So these fragments of longer molecules may not have the covalent modifications that are required for their cloning, for their adapter ligation, and they may be missed during the library preparation process. So, and we've seen yesterday that when you add these covalent modifications called fragments, you get overwhelmed by ribosomal fragments, right? So in a way, current library preparation methods are not just focused on small fragments, but small fragments with appropriate modifications, which means that tend not to be fragments of larger molecules. When you remove that bias, you actually diminish the number of reads that cover well annotated, natural, small RNAs. So in a way, this is a mixed blessing, right? In a way, we miss a big part of the picture. On the other hand, we're not overwhelmed by that huge background, of ribosomal fragments, which is not removed, prior to library preparation. Otherwise, if you're doing long RNA without poly-A selection, you would need to do some kind of ribosomal depletion and so on. So I'm not saying this is good or bad. It's just that's the way Atlas was formed, and that's the dominant technology so far.
Yeah, that's a good point. I mean, we think of big data. We have so much data to analyze, but you can't see what you're not looking for, and the the details of the RNA sequencing and which polymerases are used and how nucleic acid modifications play a role. I mean, it's maybe not as bad as chromatin, but it's complicated.
But it also speaks to RNA binding proteins. Some of them, and Eric probably can comment on this. I guess many of them may not have the appropriate modifications to be captured by our small library preparation methods. I guess when you do eClip, you do add phosphorylation, right? You phosphorylate your fragments so they're clonable.
Yeah, we do a dephosphorylation, and then the phosphorylation site is actually on the adapter when we do the ligations.
This is a good follow-up question. How much of the exRNA signal does or does not intersect with the eClip data?
An excellent question. We're actually still reprocessing all of the samples. We haven't finished yet. And so that is one of the first questions that we're hoping to ask when we finally get all of our samples processed. I haven't checked yet because it's still in work, but yeah, we hope to have an answer soon.
One thing I'll chime in a little bit about the RNA binding protein, just from the small RNA fragments. I wouldn't be terribly surprised if it's not that exciting, that it's paused ribosomes that's causing a lot of the protection of the mRNA fragments that we see. So just because of how many, how much ribosomal RNA and ribosomal RNA binding proteins we see evidence for out there. So might not be as exciting as we hope, but always worth looking.
I think that's everything I have for the RBP discussion. I reserve the right to find questions that I've missed in a pile somewhere, but because, for example, I missed a couple for Boyan. Someone asks, how long are these unannotated RNAs, and did they map, or were they mapped against microbial reference transcripts?
Yeah, so these are great questions. So when we initially looked at this, we thought it was garbage, and so we went through and tried to filter out everything that was aligning to non-human reference and so on. So once we eliminated that, then we started a more deep characterization. And so again, the typical length distribution of the small RNA precursor genes is 600 odd base pairs given the parameters that we've chosen. However, the actual clusters and the consensus sequence size of the inferred product is somewhat smaller than a microRNA. So between 19 and 20 base pairs.
So by now that we have it, how do you deal with phosphorylation status? Presumably, some of your RNA clusters consisting of multiple reads correspond to fragments of RNA. Are you taking care of phosphorylation so that you make sure you're sampling deeply and not losing many fragments?
Yeah, we've only started to really look at angles like that right now, so I don't have a definitive answer to that. And that's an excellent point.
So this is another sort of protocol question that maybe Nav needs help on. Someone mentions, ultracentrifugation based methods pellet a lot of material besides EVs, and you need to look at fractions versus total. I think they're asking did you look at total RNA, or did you fractionate it into vesicle and vesicle-free fractions in your analysis or in
And-
your sample processing?
Navneet, are you there? Do you want me to take that, or do you want to? Yeah,
So it's all you.
okay, yeah. So from what I understand, we did look at the EV specific isolates. So this wasn't just all extracellular vesicles. So we went through the standard battery of TEM, nanoparticle tracking, and immunolabeling for the tetraspanins to try and zero in on EV specific payloads. Yeah.
We have a question here, which
Sorry,
again,
and one more thing about what Boyan said. There were two ways of isolating, and we were finding similar results, right?
Yeah.
One was very smooth, which is a nanofluidics that doesn't have the the aggressive nature of isolation that centrifugation has.
Yeah.
I was about to add that the same thing, that does not, especially our microfluidic device, does not isolate free RNA.
Right. So it's in that sense, it's unusually powerful EV specificity.
Yeah.
So before we move on to deconvolution, there's a question here which is really it's a sort of foundational protocol question. And for the new people in the field, I know a lot of people on this call can answer the question. It's, are there studies on preclinical animal models? And can you discuss the effect of low volumes of available biofluid and how that affects the exRNA profile? That's like a key problem in the field.
Sorry, to whom is this question addressed?
To whomever wants to take a stab,
Oh.
but more experimentally minded people probably.
Okay.
I guess while people are thinking about whether they want to dive in, I'll just say that, because there is a low level of RNA in these biofluid, first you have to get the biofluid and then you have to pellet it down. And if you fractionate the vesicle from the vesicle-free portion, you have even less RNA. And then when you sequence that low volume of RNA, it leads to issues later in the data analysis, sort of low number effects. I don't know if Joel Rosowski's on, he might have insight into the issue. I guess if no one wants to jump in, I would tell the questioner it's a big issue and there's like dozens of papers on it. And if you figure it out, that would be great. Let's move to deconvolution. Give me just a moment. Someone asks, is there an open source program to do deconvolution, or is it an analysis that each researcher does independently on their own data set?
So I think, Emily, you can take this one.
Yeah, I can. So, I mean, deconvolution is generally performed by the researcher, but there's a lot of ways to do it. You can, so CyberSort X and CyberSort both have online tools. So you can just look them up and you'll essentially upload your data and they'll do the deconvolution there that way. The tool that Alex talked about, XDEC, and its progenitor EDEC, both are R packages. So you can download if you would like to do it in an R environment rather than online. We are also making XDEC analysis available through an online platform as well, where you would upload your samples. Deconvolution could be performed. The other methods that were talked about in Brian's talk on kind of the dream challenge, like MCP counter, timer, epic, all those other ones that came up, they're all either available as web tools, if you just search for them, or as packages in Python and R generally, if you would like to do it locally. So there are a lot of ways to do it, whether you would rather just submit to a website or have a little bit more control over some parameters for the deconvolution locally.
And EDEC and exDEC, I should add, come with vignettes, right? Small examples where you can get your feet wet and reproduce results before you apply these tools to your own data, right?
Yes, the only one I'm not entirely sure about if you would have to do it yourself would be from the first talk at A Genome Scientific, but maybe Brian knows more about that.
Yeah, so I don't know that they've released their method. I know they have, excuse me, a bioarchive paper that I think you can track down. I'll track it down and put it in the chat. I did want to add, Emily, to your comments that the baseline methods that we used, with the exception of CyberSort and CyberSort X, which, as you mentioned, are available as online tools, and after agreeing to their usage statement, you can also get a Docker instance of it. We leveraged something called immune deconv that had been used in the Sturm et al. Paper that I referenced, that has a lot of, most of those tools that we evaluated have been implemented there. I think they're all in R. I'll add that to the chat as well.
Thanks, Brian. That's helpful. Moving on, what about other normalizations that scale data between 0 and 1? Have these been assessed? I'm sure this is in reference to a particular talk, but since it was this morning.
Yeah, that was I talk about it in the context of exRNA data, where we have large variability in scale, between individual genes. Some of them may be sampled very deeply by one library preparation kit, and another version of the same kit from the same manufacturer, sometime later may downsample, tenfold the same gene, right? So the issue was how to scale, and one solution is scaling into the 0-1 range. The transformation we applied is the simplest analytically one can think of. And we played actually with variations on it, and we couldn't improve. So empirically, we tried different transformations, but none of them worked. We did actually quite a bit of trying, and we couldn't get it the simplest solution to improve the simplest solution in various contexts. So empirically, work was done. Now, whether there's a better one, maybe there is. You can always try, but we've tried a lot.
Is it a good strategy to train deconvolution algorithms with RNA-seq data abundant in mRNAs when the exRNA atlas is biased towards small RNAs like microRNA?
The deconvolution was not done just on microRNA. It was done on all the annotated regions. This is the default annotation set used by the excerpt pipeline, which included all GenCode annotations, but none of the protein coding genes or other GenCode genes had enough coverage to be informative for deconvolution.
Are these deconvolution, I think we know the answer, but are these deconvolution methods specific to mRNA profiles, or can they be used with any RNA type?
Yes, speaking of exDEC, in fact, yes, for tissues, they're used on mostly mRNA, protein coding genes. For exRNA, they're used mostly on small non-coding gene species. So in fact, they are applicable. In fact, a colleague of mine has tried deconvolution using mass spectrometry data too, and it did work. They could reproduce deconvolution of breast tumors, for example. So the scaling to 0,1 allows one to use a very diverse data types and still arrive at meaningful conclusions.
I would add that some of the methods that we looked at, some of the older methods, those still popular, MCP counter and CyberSort, the original CyberSort, were developed and trained on microarray data. So I don't know what the biases in terms of RNA species are in those platforms that they looked at, but presumably there is some bias and you'd want to look into that maybe more carefully before applying it.
Someone asks, does the amount of a given cell type in the tumor microenvironment impact the efficiency of its identification through deconvolution?
Well, we've seen the threshold studies, right? 5%. It was Brian, maybe you know the details?
Yeah, it's certainly, that was kind of a typical range. And I think that's in line with what's been reported. Both the original MCP counter and I think the CyberSort paper looked at the sensitivity analysis. And I think they reported, I think MCP was around 5% and CyberSort might have been a little lower, like 1%, but our range was from 0.2% for neutrophils to, 10% for the finer dissection of CD4s. So not too impressive, I would say. We hoped that, our study, the challenge used RNA-seq data. We thought we'd have greater sensitivity than maybe some of the early microarray studies. Maybe that was naive, but we didn't. And I'll acknowledge there's all kind of caveats in our in silico study, but hopefully at least put us in the right ballpark in terms of sensitivity.
There's a question in chat that I think I can answer. Are these deconvolution tools applicable to non-cancer data? And I think the answer is these deconvolution methods are widely applicable in fields well beyond ours. You can probably use deconvolution to study lightning patterns in clouds. I mean, and it's just a question of, domain knowledge of how to apply the math of the method into your data types.
I think the real question may be, are these deconvolution methods applicable to cancer data? In my early slides, I motivated our interest in immune profiling within cancer. The irony is our data were all from healthy donors and not in tissues. They were from PBMCs mostly. And a lot of these methods have been developed in that context, trained in that context, so they've been applied elsewhere. So the hope is that they'll translate. I think there's some disagreement in the field as to what the noise introduced by a real tissue cancer setting is.
Well, I should say about EDAC, one of my slides shows an early publication from 2016 where it was successfully applied to breast cancer. One of my slides is exDAC, which refines deconvolution of breast, but we also applied exDAC to pancreatic, head and neck, and glioblastoma. And these are all published studies. So exDAC works well on tumors.
Those are all the questions I have. Let me just point out there was a fair number of questions for Leonora Balazs. She had to go, but she has the questions and I think she intends to respond. I'll get back in touch with her about that. So I guess I would say, does anybody, if you have a question you want to ask live, unmute yourself, then mute so we don't have feedback.
I do have a question, please.
Go ahead.
This is Amin from Mr. Pittsburgh. Thank you so much for all the speakers. This is really was very informative, great. So I have a naive question. So we have this exRNA that are present in different, let's say if you look at one fluid, for example, plasma, this RNA coexists within the vesicle, outside of the vesicle, bound to proteins and so on and so forth. So what is the biological significance of this different compartment of extracellular RNA in terms of cell-cell communication or even in terms of inter-organ communication?
That's kind of the $64,000 question. If I'll summarize in case people had trouble hearing, he's asking if you have extracellular RNA inside vesicles or on the surface of vesicles or bound to proteins in all these various places, what's the biological significance? That's pretty much the question we're trying to ask. I think early on, people realized that we used to think that vesicles were garbage bags, that cells used to get rid of the toxic stuff they didn't want or need. And people realized, oh no, they're complicated signaling objects with proteins and lipids and even RNAs in them. And you know, deconstructing the biology of even one of those kinds of vesicles is not simple. There's a lot of good biology that we do know now. On the specifics of RNA in vesicles, in biofluids, I guess I'll stop there. Maybe I could ask Juan Pablo to reiterate his new his paradigm shifting findings about RNAs in fluid versus in vesicles.
Hi, Roger. I was on a phone call. I just
Sorry.
connected again. I
Sorry
hear
to
my
put you on the
name.
spot.
Sorry, can you repeat the question, please?
Yes, I'm so sorry. A questioner asked a very fundamental question, like, you know, you have RNAs in vesicles, you have RNAs in biofluids bound to proteins. What's the biological significance? And I blathered on for a bit about the field learning that vesicles are signaling devices, and thought you could talk about your paradigm shift in, you know, RNAs in biofluids and how they get there.
Yeah, so we're really not. I don't think we completely understand the biological significance of much of what is going on the extracellular space outside vesicles. It seems clear that cells die and they might release their contents to the extracellular space and biofluids, and there's a lot of RNA activity in the extracellular space. So the question is that the end of the story? Or are some, I'm not talking about the RNA which is protected inside vesicles and might eventually be transferred to other cells, but the RNA which is just free or in complex with proteins, is it just degraded? But we are seeing some fragments which just by chance or because they are protected by proteins, they resist longer, and that's what we sequence. But ultimately they will all be removed. And I think there was a question about this earlier. Are there receptors for some of these ribonucleic protein complexes or RNAs in other cells, and they can also play a role in some type of intercellular communication? And we think that at least what we are studying is their involvement regarding immunity. So we have seen that some of, and others have seen as well, that some of these RNAs, when exposed to innate immune cells, these cells will take them because they tend to uptake content from the extracellular space, and they have a lot of sensors for RNA. And for a long time, we used to think on these RNA sensors in immune cells as sensors of pathogenics, of RNA coming from pathogens, from virus and bacteria. But it's possible that our immune system is actually performing a surveillance of extracellular RNA, which can be indicative of a process of necrosis, for example. So I think that's an angle that might offer some interesting insights.
I'd also like to add that the role of microRNA in different tumors in paracrine signaling is quite well established in glioblastoma, for example. You know, the role of vesicular microRNAs in angiogenesis and immune stromal interactions, breast cancer, even metastasis in breast cancer, interaction between the cancer cell and endothelial cells, promoting the crossing of the blood-brain barrier to establish brain metastasis in breast cancer. So a lot of our knowledge comes from paracrine signaling in cancer and from, you know, immune system. Now, the difficulty in establishing firm evidence for many of these findings is that vesicles are complex. In addition to microRNA, they contain other ingredients, so pinning the effect on a particular microRNA is hard, but there's lots of circumstantial evidence in many cases and a lot of experimental as well. So the evidence is building. I believe paracrine signaling is leading the way.